Hi, everybody. Thanks for coming to this Zoom presentation. So what I thought would work best is if I give you a little bit of an overview of what I do in my lab. So I prepared 3 problems in mathematical oncology, and I'll maybe spend 15 minutes on each one. And I think that'll give you a nice overview of the kinds of things that we do in the lab. Let me just mention here that the projects that we developed over the past 5 or 6 years mostly been carried out by these 5 graduate students. 3 of them have their PhDs already, and these 2 are still working on projects related to what I'm talking about. Okay, so let me just start out with this schematic from Weinberg's book, which I think is really useful. It's a schematic of the metastatic process, and for me, it provides a really nice roadmap to define and identify good problems to work on. It pretty much goes from the development of a primary tumor all the way through colonization and metastases. And most of the projects I work on have to do with one or various aspects of this development. So what I want to talk about today are 3 projects. The first one is what I call metastatic forecasting. I'll use concepts of entropy. I'll show you how I develop Markov chain models of metastasis and how we design network diagrams of the metastatic process. The second problem I call the nonlinear dynamics of chemoresistance, and I'll talk about adaptive design of chemo schedules and how we do that. And the third project I call steering evolution using evolutionary game theory. So there I'll go into some details of optimal adaptive control of the replicator dynamical system. Okay, so before I start on the first project, let me just give you a little bit of a background. For me, a really nice paper is the 1985 paper by Spratt and Spratt that with the title, "What is breast cancer doing before we can detect it?" So for me, that's kind of a key question in a lot of what we do. And that's because before we can detect it, the tumor has to have around 10 to the 8th cancer cells in it, and its volume has to be roughly a cubic millimeter. Of course, that depends on where it's located and other things, but basically that's a ballpark estimate of when you can start detecting a tumor. So this is a diagram taken from their paper. Along the x-axis is the diameter of the tumor, and the y-axis is time. And these are different growth curves of a tumor. So the upper ones are faster, and the lower ones are slower. And a typical detection range would be around here, as I said, with about 10 to the 8th cells and about a cubic millimeter in size. And the question is, what is it doing in this region? And we want to push that yellow rectangle to the left as far as possible. So one of the things that we do in our lab that I'm not going to talk about today is we focus on the mechanics and biomechanics and deformability properties of circulating tumor cells. So Peter Kuhn at USC has a lab where he looks at circulating tumor cells and circulating tumor cell clusters. And we take those images and we develop deformable immersed boundary methods of these cell clusters moving through the bloodstream and diffusing chemicals that induce blood clots. So I'm not going to talk about that today, but that's just one of the tools that we use. So basically, to push that rectangle to the left, we need better detection, and we need things like fluid biopsies. Those can help push that rectangle to the left. But what we do are mathematical models and computer simulations that kind of fill that gap as well. One more thing. So up on the upper right here is a typical tumor growth curve, which is a Gompertzian curve or an S-shaped curve. And that comes from Laird's paper in 1964, where she took lots of different kinds of tumors from mice and rats and humans and normalized them and put them all on the same plot and then fit them to a Gompertzian curve. And so for me, that's one of the nicest experimental curves I've seen. Um, and one of the things that we've developed over the past couple of years is a stochastic Moran process model that kind of mimics a lot of the features of this. And so that's an S-shaped curve. That's a realization of our stochastic problem model. And the thing is, when you have an S-shaped curve, you have inflection points. And so you have one inflection point here in the red, and then you have two inflection points of the derivative of the Gompertzian. And so for me, that breaks up this curve into different regions. So to the left of the blue inflection point is what I would call the subclinical region, where the tumor is not yet detected. And then between these two inflection points would be, generally speaking, the clinical regime, where the tumor typically gets detected. And then to the right of the second blue inflection point is the lethal burden. And as I said, you're up in this region here with 10 to the 8th plus cancer cells. And the thing is, from an oncologist's point of view, early diagnosis would be somewhere around here. And that would be typically the regime in which there's linear growth of the tumor. But the problem is the first driver mutation typically takes place way over here to the left. And there's probable dissemination into the bloodstream, let's say over here, way before the tumor is detected. Okay, so that's basically the challenge that oncologists face. So let me now start describing problem 1, which is metastatic forecasting using entropy methods, Markov models, and network diagrams. Okay, so the first point that I want to make is stochastic does not mean unpredictable. Now, I think probably a lot of people in the audience realize this, but there might be some people that don't. And so let me just, and it also sets a stage nicely for the kinds of models that we built. So let me walk you through that. So imagine coin flipping. So consider a sequence of coin flips with a weighted coin. So there's two possibilities. You either land on heads or you land on tails. So there's two states. And let's say P is a probability of heads and 1 minus P is a probability of tails. So in a fair coin, P would be 1/2, and you would get a sequence of, in one realization, you get a sequence of H's and T's, let's say that looks something like this. And roughly speaking, after a long enough sequences, you start converging by the law of large numbers to roughly 50% there's 50% of them are H's and 50% are tails, are T's. Okay, but in an unfair coin, let's say the probability of heads is 0.1, then you're going to get far more T's than H's, and in the long run, you're going to get roughly 90% of the elements in this string are T's. So both are stochastic processes, but the point that I want to make is that the unfair coin sequences are much more predictable, and that's because all you need to do is just predict tails every time here, and you'll be right 90% of the time. Whereas here, if you pick tails every time, you'll be right only 50% of the time. All right. So the way to quantify that in a scalar quantity is by using this formula for entropy, which probably some people are familiar with. So if P1 is the probability of heads and P2 is 1 minus P1, the probability of tails, then there's going to be two states here. That's why I'm summing from I equal 1 to 2. And if we plot this function as a function of the probability of heads, it looks like this, okay? So the minimum entropy states, where the entropy is 0, would be down here, and that would be either a full string of H's or a full string of T's. The maximum entropy state would be the fair coin, where it's 0.5 probability heads, 0.5 tails, and you get roughly half H's and half T's in a string, a string that's long enough. But somewhere in between, where you have an unfair coin, and it's, let's say the probability of heads is somewhere between 0 and 1, but not 0.5, then you're going to have some sort of string of H's and T's, and that's going to be some intermediate entropy state, less than the maximum, but more than the minimum. And the point that I want to make is that high entropy sequences, the one up here, is less predictable than the low entropy sequences. Okay, so that's a nice scalar quantity that allows you to characterize that quantitatively. Okay, so we've got this sequence of coin flips here, H's and T's, and it's a long, let's say it's long enough. And what I want to do now is talk about an analogous sequence um, of the metastatic process. So I'm going to focus on breast cancer, and I want to imagine one realization, which would be one patient that starts out with breast cancer, with a breast tumor, and their first metastatic tumor goes to the lymph nodes. The second one, then a second one appears and goes to the bone, and then a third one appears and goes to the liver, and then the adrenal gland, and then the patient dies. So I'm going to view that as a metastatic sequence. And what we've done here is we've taken data, which is autopsy data from the 1920s. So this is baseline data. These patients were untreated. And there's something like 3,800 patients, and they have multiple METs, and so the data is organized according to the location of the METs, and then we normalize that data so that the area in this histogram under these bars is 1, so that we can view this as a probability distribution, okay? And there are 30 possible sites, and this is for a whole bunch of different kinds of cancers, and I'll split them apart in a minute. But you can view this as a weighted coin with 30 different sides, and the height of this histogram is the probability of landing on each of these sides, okay? So we're gonna then compute the entropy of all the cancers taken together by summing from 1 to 30 using that same formula where PI is the probability of a metastatic tumor at site I, as I said, where I goes from where N is 30, because those are the 30 different sites in this data set. And so this is what the distribution looks like for all cancers taken together, and the entropy is 2.714. And that is a signature of how quickly that probability distribution goes down to 0. All right. Then we broke it up into different kinds of cancers. So up here, we're starting with the highest entropy cancer, breast cancer, with an entropy of 2.78, and then lung cancer, slightly lower, 2.745, and then all cancers taken together, so breast and lung, are higher entropy, meaning less predictable than the average. And prostate cancer down here is a very low entropy cancer. And you can see that immediately just by looking at how steeply this distribution drops off. And what these are in these insets are Markov diagrams, reduced Markov diagrams that correlate. The complexity of these diagrams correlates with the entropy associated with the cancer type. And I'll tell you more about how we get those Markov diagrams in a minute. So what we did was we took 12 different cancers and we ranked them in terms of their entropy. And so we've got high entropy cancers, which are skin, breast, kidney, and lung. And those are all above average. The all cancer is right here. We've got medium entropy cancers, which are a little bit more predictable than the high entropy ones. And those would be stomach, uterine, pancreatic, and colorectal cancers. And then we have low entropy cancers, which are the most predictable. And that would be cervical, ovarian, bladder, and prostate cancers. And the entropy associated with those, as I said, correlates with the complexity associated with the network diagrams of those cancers, which I'll talk about in a couple of slides. Now, I'm not making any statement about how hard these cancers are to treat. That's obviously not part of this analysis. I'm just talking about the predictability of a model like this. Okay, so how do we get these Markov diagrams? So now I'm going to turn towards longitudinal data. So these are all treated patients. And so the data set that I'm going to show you comes from Sloan Kettering, collected over 15 years, and I'm going to focus on breast cancer patients. And I'm going to show you how we take their longitudinal data and turn them into Markov models. So you're probably all familiar with weighted directed graphs. These are networks with nodes on them. So I've just listed a simple, I'm showing a simple one here with 5 nodes, and they have edges, and those edges have directions and weightings. That would be a typical weighted directed graph. And those kinds of things have been used in different biological contexts and medical contexts. For example, I'm showing here a brain neural network. I'm showing a human disease network, and I'm showing a microbial network in 3 different, developed by 3 different labs. And what I'm going to show you is our metastatic cancer network. And this one I'm just going to show you here is lung cancer. So we have these 30 nodes where metastases can show up, and we have directed weighted edges that join the two, any 2 pairs in this diagram. And as I said, the nodes in that network are the locations in the body where a metastatic tumor could possibly appear. And then since that first diagram was too complicated, we then reduce these diagrams by just taking the top, let's say 36 in this case, % of the information contained in that graph, which is typically good enough, and draw reduce Markov diagrams that look something like this, where the primary tumor is here and the metastatic locations are around from 12 o'clock all the way around in reduced probability. And the blue sites are called the sponge sites, and the red sites are called the spreader sites. And those are characterized as either having a higher probability in than out, in which case they're a sponge site, or a higher probability out than in which case they're a spreader site. And if they're not colored, then those probabilities are too close to really characterize them one way or the other. Okay, so the first thing we do before we even make Markov diagrams is we just look at the data. So this is what we call tree ring diagrams. So this is, we take the longitudinal data and we just display it in this way, where these are, we can think of these as tree rings. These are all breast cancer patients. So the inner ring is all primary breast tumor. The first ring out from that is the first MET, and the sector size gives the probability of the location. Okay, so here we've got around a little less than 50% of the patients have their first MET to the bone, and then a little less than that to the chest wall, lung, lymph node, liver, and so forth. So that's the first MET. And then this next ring out is the second MET, and again, the colors indicate the location. Here's the list, and so forth. And so this gives you sort of just a nice tree ring display of all the patients that we have, the breast cancer patients that we have, in this case, 446 patients and 310 distinct pathways. No modeling here, just data display. But the data display allows us to see these patterns develop in time. So this is a 10-year progression of these tree ring diagrams. So now we're on year 7, year 8, year 9. And so you can see the patterns develop over a 10-year period, and these patterns emerge over time. And these are what I would call spatiotemporal pathway progression diagrams. All right, so you can also, of course, split these up into molecular subtypes, which we're doing here for breast cancer. And then you can look at the differences in the patterns. For example, here's ER plus HER2 negative patients. Here's the ER negative, HER2 negative, and HER2 plus patients. And you can see that the patterns can be quite different in these different molecular subtypes. Part of that has to do with the fact that there's different numbers of patients in each of these groups. For example, there's 218 patients in this subgroup, and there's only 62 patients in this. So yes, part of it has to do with that, but there's also information that you can pull out of these more carefully. All right, so from those spatiotemporal diagrams, we can then start putting together Markov chain models by computing the transition probabilities from each site to every other site. And that gives us then the ability to construct Markov processes. And here would be the full network structure of the breast cancer cohort. So the primary tumor is always at 12 o'clock. And then as we go around, we have the metastatic sites, and bone would be the highest probability, and so forth as we go around, lower and lower probability. And the the way this software works is that the thickness of these lines is the transition probabilities from outgoing to these various sites. So it's a bit crowded. You can't really see much, but for example, you can ask all kinds of questions on this live website. For example, you can look at just the paths from the breast. So this would be the first transition from the primary tumor to the Mets. And you can see that these are the first Mets that we have in our data set, and from higher probability to lower probability. You can ask questions about, for example, if you want to target a specific site where there's a Met, like the bone, you can ask what are the paths that go into the bone. And so we can highlight those in the diagram. These are all the paths leading to the bone. Or you could ask questions like, what is the last Met before the patient dies? Because we have deceased as one of our nodes here. And those are all the paths that lead to that final site. Okay, well, as I said, those diagrams are a little busy. So what we do is we reduce the diagrams by only taking about a third, let's say 35% or so, of the information that those graphs contain. And that, as I said, contains most of the information. So here is the reduced Markov diagram for all the breast cancer patients taken together. And bone is always at 12 o'clock. That's the most probable metastatic site, first metastatic site. And then we broke them up into molecular subtypes. So ER plus, HER2 minus, ER minus, HER2 minus, and HER2 plus. And then it allows you to look at the differences of the diagrams associated with those sites. Okay, so you can see here that the HER2 negative group has, for example, I'm just highlighting one difference, has lung pleura as a sponge site, fairly high up, fairly probable, but not the most probable. Whereas this group, the ER negative, HER2 negative, has the lung pleura site much less likely down here, although they are both sponge sites for that group. On the other hand, for the HER2 positive group, the lung pleura site is much, much more probable. It's much closer to 12 o'clock, and its character totally changes. It's a spreader site. So that's just one example of many that is different about the HER2 positive group than the HER2 negative group, for example. All right, one last point that I want to make here, and that is that the complexity of the network diagram is very, very highly correlated to the entropy number that I showed you earlier. So for all of these cancers that I had showed you before, we're plotting here entropy versus the something called the graph conductance, which is a measure of how fast a random walk model converges to its steady state, which you could think of as the Markov mixing time. So these are very, very closely linearly correlated. The higher the entropy, the longer the model takes to converge to its steady state, which gives you an idea of the complexity of the Markov diagram. All right, there's a lot more to say about that, but let me move on to problem number 2, which is focused on the nonlinear dynamics of chemo resistance. And I'm going to talk about adaptive design of chemo schedules. Okay, so what I want you to think about now is thinking about the tumor as an adaptive, co-evolving ecosystem of different kinds of cells, just the way you would view a jungle with different animals co-evolving, or a fish population in the ocean with different fish co-evolving. And with respect to metastasis, I want you to think in terms of ecology and evolution, and think of dispersal kinds of issues, like how the dandelion seeds disperse in a similar sort of a way in which metastases spread in the body. And I want to point you to a quote from an important paper in 1976 by Noel, where he says that more research should be directed towards understanding and controlling the evolutionary process in tumors before it reaches the late stage seen in clinical cancer care. And I want to focus on that because for me, those are marching orders. That's what we try to do. Okay, so what I'm showing here is now I'm focusing on prostate cancer. So I'm showing the PSA level on the y-axis, which is a biomarker, let's say a surrogate for, let's say, tumor size, and the x-axis you can think of as time. And this is for a group of different patients. What you see in all of these patients is you see when they have chemotherapy, there's regression of the tumor in the beginning, and then eventually the tumor regrows. And the question is why that is, and why is that so robust? And I want to point out here that these are three different kinds of chemotherapies, prednisone, actually, I'm not really going to be able to pronounce those too well, but different kinds of chemotherapies for prostate cancer. But the thing is that response is very robust and persistent across patient populations and treatment protocols. So it's what I would characterize as chemo resistance via competitive release. So what is that? So let me describe it this way. So let's coarse grain the tumor, and let's say we can't keep track of every individual cell. So what we're going to do is group the cells into, in this case, three different groups. We've got healthy cells, we've got cancer cells that are resistant to treatment, A, let's say, and then we have cancer cells that are sensitive to treatment A, okay? So think of it this way. So in the beginning, before treatment, you have a domination of the sensitive cells in the tumor, and that's what's driving the tumor growth. Those are the red cells. And then you have a small little population of the green cells that are the resistant population. And so you hit the tumor with chemotherapy, and that kills a big proportion of the sensitive cancer cells. And so you reduce the number of red cells quite a bit. You don't really reduce the number of green cells because those were resistant to treatment. But now what you've done is you have changed the playing field. And so now the resistant cells are able to compete better because there aren't as many sensitive cells, and those two populations were competing against each other for resources and oxygen and nutrients and so forth. And so what happens is they've been released from the competition, and then they start to grow, and your tumor regrows. And now, and that's the tumor volume getting bigger, but now your tumor is made up mostly of resistant cells, and so the chemotherapy no longer works. And that's seen here in a different way, in a fish plot, where time is along the x-axis. So the first driver mutation occurs there, and then the sensitive cell population grows, and then you have very good initial response to chemotherapy, and then the resistant cells start to then grow because they're released from the competition and they take over. And that is a very common story. All right, so how do you model something like that? So these are the replicator dynamical system, and in this case, it's three groups of cells. We've got the healthy cells, the sensitive cells, and the resistant cells. These are frequencies. So the sum of the three is 1. So it's just the frequency of these cells in the tumor. And FH, FS, and FR is the fitness of that particular subpopulation. And the bracket F is the average population fitness. So what this system says is if the fitness of the subpopulation is above average, then that subpopulation is going to grow exponentially. And if the fitness of that subpopulation is below average, it's going to shrink exponentially. And that's basically the replicator system. But the thing is, these are all coupled. And they're coupled based on how you define the fitness landscape. So here we're showing the fitness landscape. Typically, the fitness landscape would be just defined in terms of a payoff matrix, which in this case will be a 3x3 matrix that defines the interaction of these three cells. But we're also using a parameter here, which we're going to use these parameters are going to be functions of time, and those are going to be our chemotherapy treatment schedules, okay? So, and then the average fitness is given by this formula. All right, and as I said, we're introducing a chemotherapy time-dependent function, which defines our chemo schedule. And basically, the chemotherapy parameter or time-dependent function adjusts the fitness landscape associated with the subpopulation. So when you turn on chemotherapy, then it reduces the fitness of the sensitive cells, but it doesn't affect the fitness of the healthy cells or the resistant cells. And so what that does is it takes you on some path in this trilinear phase space: sensitive, resistant, healthy. All right. So as I said, CFT is the drug concentration profile. It's going to be a time-dependent control parameter. And whenever we compare treatments, we are restricting it to the same total dose. So the integral under CFT is going to be the same whenever we compare treatments. That's our constraint. Different kinds of dosing schedules would be these. The typical oncologist would use maximum tolerated dose, MTD, which is just a sequence of pulses at a fixed level, independent of how the tumor responds. So it's all pre-scheduled. There's nothing adaptive about it. LDM is low dose metronomic. That's also somewhat common, where the oncologist gives a very low but continuous dose of chemotherapy. And again, to compare these schedules, the areas would be the same. Or you could do a ramp up and ramp down. These are all pre-scheduled. Adaptive doses, on the other hand, could be anything. The CFT, in principle, could be any of these profiles. But as I said, the total dose of the adaptive should be the same as any of these in order to compare them head to head, which is what we do. All right, so first of all, I want to show how competitive release works in these models. So this is our trilinear phase. This is our trilinear coordinate system. And we start somewhere here, and this is what happens when there's no chemotherapy. So when there's no chemotherapy at all, the sensitive cell corner is going to be the asymptotically stable state in this model. And so every trajectory goes to this corner, which means the tumor saturates when S is 1 and H and R are 0, and the tumor saturates with sensitive cells. That's with no chemotherapy. On the other hand, when the chemotherapy is above a certain threshold that I'll tell you about in a minute, all the trajectories go to the resistant corner, which is the evolutionary stable state, ESS. And that, in essence, is competitive release. It changes the ESS of the system. All right, so how does that work for more finer tuned differences in the chemotherapy? By the way, we're not doing time dependent yet. I'm just showing you right now when you have a constant chemotherapy value. So when the chemotherapy value is too low, like at 0.3, then all trajectories converge to S. That's the red basin of attraction here. When it gets a little higher, the basins start to intertwine, and there's a basin of attraction in the resistant corner, and there's a basin of attraction in the sensitive corner. And as C gets larger and larger, the basin of attraction in yellow, which is the converges to R, gets bigger and bigger until when C is 0.6, the entire triangle is in the basin of attraction of the resistant population. So here R would be an unstable state, and here when C is 0.4, it is an asymptotically stable state for tumor makeups, frequencies that start in the yellow regime somewhere. All right, well, you can look at that even more closely by what I'm showing you here is now C going from 0.33 to 0.48. And now I'm also plotting the null lines, which also gives good information about how these trajectories are going to move as you increase C. So these are more detailed on the basins of attraction and how they change. And for example, you can use the null lines to drive trajectories into different corners. So here would be an example of a case where the maximum tolerated dose schedule goes to the sensitive corner, Whereas when you do adaptive therapy, by switching the chemotherapy off and on at just the right times, balanced between these null lines, you can linger in this region for quite a long time and keep the cells in competition without ever saturating the tumor. So you can drive it in a certain direction by switching the chemotherapy off and on, and keep it away from that S corner, or and keep it away from the R corner. And here would be another example of switching the chemotherapy off and on in order to trap it between these null lines, keep it away from those corners. You could have it go up, you could have it go down, and so forth. So there's a lot you can do once you adaptively have your C of T as your control. So let me show you this before I talk about adaptive control. So what the model shows is that if you have different values of C from 0.3 to 0.55, this is the proportion of the areas in that triangle of the basins of attraction. So as C goes up, the basin of attraction of the sensitive cell population goes down, and the basin of attraction of the resistant population goes up, and they cross that are a little bit bigger than 0.4. All right, so you can use that information to your advantage. So think of the following. So here's the situation when there's no chemotherapy, and here's the situation when C is 0.7. So what you can do is you can lay those trajectories on top of each other, and that allows you to choose a closed loop. Here, for example, one of the closed loops that we call evolutionary cycles. And all you need to do in order to stay in this closed loop forever, at least in this model, is to calculate the times that you need in order to switch from one trajectory to another. Okay, and so here would be an example of what the adaptive therapy would look like if you were able to compute, if you were able to use this model. Once you have this model, it's very straightforward to compute these times. And so you first do the adaptive schedule, and then you compute the area under these curves, and then you compare that to a maximum tolerated or a low dose metronomic with the same area, same total dose. All right, I just want to point out that this kind of a technique is known in the astrodynamics literature kind of as a Hohmann transfer method, where you're on a trajectory. Let's say you're a satellite or a rocket, you're on this trajectory, and then you want to get it to this trajectory. So you have to use some control where you turn on the propulsors and basically get it from one trajectory to another. So this would be one trajectory, this would be the other trajectory, and then there's a transfer orbit. And you just have to calculate the times at which to turn on the controller and turn off the controller. And then when it's off, it's on this orbit, and before it's off, it's on that orbit. So people have used this to design better orbits, to compute time optimality. And in this case, you might have other constraints like the total dosage. In the astrodynamics case, you would have propulsive energy. So in this example, it requires 3 values of C and 2 transfer times. All right, so the same kind of thing can be used here. So this would be a picture of a closed orbit that just goes between A and B for 2 different values of the chemotherapy, switched off and on at just the right times. And what we're doing is we're comparing the results of that adaptive situation to maximum tolerated dose, which is in purple here, and the low dose metronomic, which is in green here. And what you can see is both maximum tolerated dose and low dose metronomic with the same total dose will go to the resistant corner. So the tumor becomes resistant, whereas in our adaptive schedule with the same dose, we just stay in this evolutionary cycle forever. And we never go to either the S or the R corner. Okay, here are the schedules as they compare, and here I'm showing the fitness of the tumor using these different schedules. All right, well, the whole story becomes doable but more complex when you have multi-drug scheduling, and so we're working on models for that. So now we have C1 and C2. We have 2 different chemotherapy schedules, And we have the ability to have the drugs interact synergistically. And we have this parameter E. If it's positive, it's synergistic drug interactions. If it's negative, it's antagonistic. And if E is 0, then these are just additive. Okay, so now our model is we have 3 populations. We have a sensitive population, which is sensitive to both drug treatments. We have C2, which is resistant to drug 1, and we have C1, which is resistant to drug 2. Okay, and so when there's no chemotherapy, just as before, so now our corners are S, R1, and R2, and when there's no chemotherapy, just as before, every trajectory goes to the S corner, meaning the tumor saturates with cancer cells that were sensitive to both treatments. On the other hand, when C2 is large enough, so when one of the drugs is turned on, then all of the trajectories converge to the R1 corner because these are the cells that are resistant to drug 2. And when C1 is on, large enough, then all of the trajectories converge to the R2 corner because these cells were resistant to drug 1. And so we can use that information and toggle and design trajectories using all that information. So to talk about this synergistic, additive, and antagonistic behavior, it's our model has our fitness parameters in a formula that looks something like this, where the fitness is reduced by C1 and by C2, and a cross fitness, C1 times C2, which is multiplied by this parameter. And as I said, when E is positive, it's synergistic, meaning they're both helping each other. And when E is negative, they're actually combating each other, the drugs. And when E is 0, they're just additive. Okay. All right, so let's look at what happens. So here we've got the structure of the basins of attraction when E is negative, so they're antagonistic drug interactions for 2 different values of the chemotherapy schedules, C1 and C2 of the drugs. Here I'm showing when the interaction is additive for the same values of those chemotherapy parameters. And here I'm showing when they are synergistic for those same values of the parameters. So what you can see from that, I'm just showing them all together here, is that whether or not the drug interactions are synergistic or antagonistic or additive is important in terms of understanding how the phase structure changes, which is going to be important in terms of designing these adaptive evolutionary cycles. But you can still do it. So here would be an example of an evolutionary cycle with the two drugs. And so what we do here, I'm showing an evolutionary cycle between O, A, and B. So it just is trapped in this little triangle here, which would be up here. And this is the schedule, the time-dependent schedules of C1 and C2 in order to trap it in this orbit. And so what we've done is we've compared patient data and actual and these kinds of schedules to see how they could do in principle. And they work much better. So the dots are the patient data for one example, the prostate cancer patient, where you have the tumor responds nicely to chemotherapy, and then because of competitive release, it starts to grow again. And what we do is we compare that with different kinds of treatments. For example, therapy 1, therapy 2, and therapy 3, which basically are maximum tolerated dose therapies, don't work so well. Whereas our evolutionary cycle, our adaptive therapies work much better in the sense that they keep the tumor from growing too large. All right, and we've done other things. For example, we've taken patient data in some fixed time interval here, and we've tuned our models based on that data, and then we've moved forward in time after we tune them to patient data, and we can see we can stay trapped in evolutionary cycles for quite a while, designing the adaptive schedule with multi-drugs. Here's Lupron and abiraterone. for prostate cancer patient. So there's a lot to say here. This would just be an example. This blue schedule basically is being trapped in this evolutionary cycle. All right. So let me now move to problem number 3, which is steering evolution using evolutionary games. So I want to now talk about optimal control, optimal adaptive control of the replicator dynamical system. All right. So just to remind you, standard control theory methods are typically based on linear inhomogeneous systems that look like this, some initial data, and you have a matrix, and you have an inhomogeneous term, and you have a variation of parameters formula, which solves the system exactly. And based on that variation of parameters formula, you can use all kinds of tools in linear control theory, like controllability, the bang-bang principle, time optimal control, Pontryagin maximum principle, dynamic programming, just to list some of the standard things that you can bring to bear on problems that are of this form. The problem is the replicator dynamical system that I showed you before is a nonlinear evolutionary system with this payoff matrix, and it's just not in that form. So none of these ideas carry over directly. So what I'm going to show you is some results in a 2-component model that I will that we can generalize to higher dimensions in a pretty straightforward way. So here's our replicator dynamical system, and as I've told you before, A is our payoff matrix that defines the kind of evolutionary game that's being played. And I'm going to show you an example of the hawk-dove evolutionary game, which is one of the standard evolutionary games where the hawks are the aggressors and the doves are the passive population. And this, again, frequency-based, so they sum to 1. And what I'm going to describe, so basically the baseline payoff matrix is the hawk-dove payoff matrix. And then what we do is we add a control matrix to that with off-diagonal bounded controllers. And that adjusts the payoff entries of the matrix, which basically means you're shaping the fitness landscape of that system. But you need to shape it in the right way. So the frequency, so here are the bounds. So x1 goes from 0 to 1, x2 goes from 0 to 1. And our controllers are normalized so that they can go from -1 to 1, but in principle, they could be any time-dependent schedule whatsoever. And the baseline model is a hawk-dove evolutionary game, which means that there's an intermediate fixed point of that system, which is asymptotically stable, which means that the hawks and the doves in this game, if there were no controller, they would gravitate towards some coexistence. Neither one of these populations is going to dominate because those are both unstable fixed points. And so what we ask in this problem is, can we design controllers in order for us to, for example, minimize aggression or maximize aggression, like push this fixed point to the left or to the right over a finite amount of time with some optimal controllers. All right, so that takes us to this 2x2 diagram, which are the entries of the off-diagonal elements of the payoff matrix. And in principle, always start out in this example with hawk-dove. But with time-dependent controllers, that means that we can go along some path in the plane and we end up over here. So each path defines a sequence of games. And the question is, what path minimizes or maximizes aggression after a finite time? All right, so as I said, the uncontrolled problem where the controllers are turned off, everything gravitates towards this mixed state, which we've cooked up to be 1/3, and that's what the dynamical system looks like. But turns out that the optimal control is a bang-bang schedule. So we start at hawk-dove, we then go to deadlock, we then go to leader game, we then go to prisoner's dilemma game, and then we go to game number 9. And so that's just bang bang, meaning we're turning these U1s and U2s off and -1 and +1. And so what I'm showing you here is just an example of one initial condition. So let's say we start out with the aggressor population at 0.08, and we first go to the deadlock game. So we start here. The deadlock game pushes us to the right. We then switch to the leader game, so that pushes us to the right a little more slowly. We then go to the prisoner's dilemma game, and that pushes us to the left. And then we go to the game number 9, and that pushes us to the left more. So that would be an example of a optimal schedule. So here are the control schedules. The blue curve is the maximum that you can achieve for the aggressive population. The red curve is the minimum that you can achieve for the aggressive population, and the dash curve would be the uncontrolled hawk population. So with the blue schedule up here, going from -1 to +1, we can achieve the maximum, and with the red schedule, we can achieve the minimum. And the schedule, the optimal schedule that you get by Pontryagin's maximum principle and computing it, it depends on the initial condition. So for example, if you have a large population of hawks compared to doves, so here the hawk-dove ratio is 9:1, here the hawk-dove ratio is 1:9 initially, you have a completely different schedule. But you can still get a maximum and a minimizer. And the nice thing is these schedules basically then define upper and lower bounds on all possible schedules that control the hawk population. So all schedules would produce a result that would be somewhere between the max and min. And so you can then ask yourself what happens over longer times. So what I showed you before was just one time cycle from 0 to 1. We've normalized it. And now what we do is we go from 0 to 1, we optimize there, and then we look at where the population is at the end of that time, and we produce our new schedule to go from 1 to 2, and then we produce a new schedule and a new schedule and so forth. And it turns out that there are absolute minimizers that you can't ever get under, and that there's absolute maximizers that you can't ever get over. So there's a limit to how well you can do, depending on what your initial population is. There's an absolute maximizer and an absolute minimizer. And so these we would call theoretical bounds on the maxes and mins that can be achieved. And so we're currently using this framework to study vaccine incentive scheduling and design for example, COVID vaccines, and ask the question, is there a theoretical limit or threshold to how well incentives could work? So again, incentives would just be time-dependent schedules that change the fitness landscape, meaning the entries of the payoff matrix between the two populations, vaccinated and unvaccinated. So we're using data from the CDC, and we are designing optimal schedules and to see if we reach these limits. Okay, so those are the 3 problems that I wanted to share with you. Let me just summarize here by saying that the goal of our lab is to add value to longitudinal medical data by building dynamical systems models that are predictive and actionable. And the tools that we generally use in our lab would be these kinds of tools: Markov chain network analysis tools, low dimensional representations, evolutionary game theory models, adaptive and optimal control theory, entropy-based methods, uncertainty quantification and Monte Carlo methods that I didn't get to talk about much, and stochastic Moran birth-death processes that I also didn't get to talk about too much. So these are the students, some of whom are still working with me, some of whom have graduated, that have worked on these kinds of models over the years, and this is the where we get our support. So thank you very much.